Welcome to lesson 6.10. This is our last lesson in unit 6 on protozoa, and in this lesson we'll look at soil protist ecology. We'll examine the effects of soil protists on the rhizosphere, so how they affect individual plants and the interaction with plant roots. Then we'll look at how the presence of protists in the soil affects plant community structure. We'll examine protist role as bioindicators of soil health, as well as their powerful role in bioremediation. So this is where organisms clean up pollutants in soil, water, and other parts of the ecosystem. And then we'll end this unit with a summary of what we've learned, as well as look at future research that still needs to be done in order to fully understand the important role of soil protists in the soil food web and the soil ecosystem. Alright, so let's jump into our final lesson of Unit 6, Lesson 6.10. So first of all, let's discuss protozoa in rhizosphere ecology. How do they fit into this rhizosphere ecosystem? The rhizosphere is obviously a micro-ecosystem because it's concentrated to the root, specifically to the root part where the root exudates come out from the root. So particularly the root tip, and this influences soil chemical, physical, biological and biochemical processes. So these differ to other soil parts, and specifically the root zone microsites create this ecosystem through root growth, through water and nutrient absorption, respiration, so the breathing mechanism of the organisms as well as the roots, rhizosphere deposition, so all the chemicals that the plant deposits, as well as the cells that it sloughs off and the dead root debris that is deposited. There are specific biochemical substances within the root zone microsites and very specific soil biota, so the different organisms that live around that root zone microsite. So all of those particular characteristics are specific to a particular plant and its root system. The physical and chemical properties of the root zone or the rhizosphere is very different to that in soil without plants. And the rhizosphere microecology is also different to the whole soil ecological system. There are 34 times more microbes in the rhizosphere compared with bulk soil, so soil that is further away from the root. And there are definitely hotspots that are influenced by living roots and organic matter that produce several million organisms or microbes per gram of the soil that comprises the hotspots. Different plant species and the stage of growth of that plant also strongly influences the microbial community, and it produces microbial community variation. That's because, once again, all of these specific mechanisms related to specific roots govern the microbial community, and as the plant changes, as it grows, that also influences what happens in that rhizosphere microecosystem. So here we can see how the rhizosphere ecology is influenced via the soil environment, which then depends on abiotic factors such as geography, climate, soil type, moisture conditions, temperature, etc. And this influences then the bulk soil microbiome, so all the microbes that live around the root zone, but also the rhizosphere microbiome. And the rhizosphere itself, or the plant itself, produces not only food for the microbes that it wants to have living around itself, within that rhizosphere or within itself, but it also produces chemical signaling to attract particular microbes, because these microbes will recognize that chemical signal and recruitment will occur. And of course, that recruitment then depends on the competition and the colonization capabilities of these microbes. So all of this plays a part, and all these things influence each other. And of course, whatever the root provides, the organic acids, the small molecules and the hormones that the plant produces to stimulate the growth and attract the colonization of its root system by different microbes, is very much dependent upon all of these factors. So it's just like a loop. Whatever comes at the plant from the outside influences what happens underground. And then what happens underground very much influences what happens to the plant, and therefore what happens to the larger atmosphere around the plant. It is all very much cyclical, and each of these impact on one another, and so it is just a system. We need to understand the rhizosphere ecology as part of the greater system, the global system. Really, we're looking at micro scale to sort of macro scale, to like an individual plant, to the whole forest or paddock, to the whole biological system of the biosphere, and that includes climate. We need to learn to appreciate and understand that the individual soil microclimate has a huge impact on the global climate, because each gram of soil has the potentiality to fully impact in a positive way the above ground biosphere. So how we manage soils at that rhizosphere level and the biodiversity of organisms that we allow to proliferate has huge, profound impacts on the large-scale ecosystem of the entire Earth biome. This is why it's so important to understand these processes and then learn how to manage them in order to affect biodiversity, climate, food security, and all the other components essential to our very existence on a global scale via these microscale ecosystems that exist in soil around each and individual plant. Soil protists, or soil protozoa, affect individual plants via the root zone rhizosphere. The very act of predation on bacteria and the bacterial remains actually produce hormonal active substances, which affects the plant on a hormonal level. The protozoa themselves actually create hormonal precursors, and these hormonal precursors and the microbial hormonal active substances increase root complexity, lateral root growth and expansion, the surface area of the root system, and that increases water and nutrient uptake. So protozoa are absolutely essential predators of that microbial community around the plant root that stimulate plant growth on that very much chemical level, just by interacting with their prey, with the bacteria and other organisms that they prey on. Protozoa therefore increase fine roots and root tip biomass, and this leads to multi-branch root structure, increasing finer roots. And of course, when this happens, that means there is a greater surface area for absorption of nutrients and water via the roots. And of course, that depends on the plants, as some plants also have their mycorrhizal fungal partners on their roots, which further extends the root system and increases its surface area, and that would also be influenced by protozoa grazing activity. For example, wheat seedlings have increased lateral roots and strong fibrous root system production, which leads to increased nutrient uptake when protozoa are present. A genus of amoebae called Acanthamoeba influences microbial activity and community composition, and therefore increases auxin production, and this is also known as indoacetic acid, which is a plant growth stimulating hormone. Protozoa also increase plant growth promoting rhizobacteria, which we have covered in our unit on bacteria, and this leads to the production of hormones and increase in root growth. Also, when protozoa feed on bacteria, they increase nitrate presence in the soil, and this has hormone-like effects on the form and the shape that the roots take on, also increasing lateral root elongation. So we can see how the presence of these particularly bacterial predators leads to hormonal effects on plants. So while plants produce all these hormones themselves to allow them to benefit their growth, the presence of bacterial grazers and the type of bacteria in the rhizosphere also stimulates plant growth, as plant hormones are released through these ecological interactions of all these organisms working together. In addition to affecting individual plants, soil protists also affect plant community structure and diversity, and in fact also the multi-trophic above-ground interactions. So how the plants then interact with, for example, the herbivorous animals that feed on the plants. Species interactions within soil or any other ecosystem determine the ecological structure and function of that ecosystem. So on a micro scale at the rhizosphere level, plant microbe and the microfauna which feeds on those microbes, these relationships are very much species specific. So the plant relationship to the microbe and the microbe relationship to the microfauna which it feeds are very much species specific. So it's a very specific interaction that occurs between all the players within the soil. So therefore the microbial population which the plant cultivates around itself, around its root, and grazing microfauna have huge effects on plant growth. And these microfauna relationships promote some plants to grow and disadvantage others. And this influences plant diversity. So microfauna, microbial rhizosphere communities, so the interaction of soil protists with the microbial rhizosphere communities, therefore impacts plant community structure, diversity, and therefore the herbivores that plant community feeds. This interaction between microbes and microfauna also influences chemical composition of plants and therefore nutrient content of plants as well. And this determines the herbivorous animal communities and also their antagonists, so the organisms that, for example, parasitize or feed on these herbivorous animals, be they insects or larger animals such as mammals. This influences the soil food web trophic cascades, and we have looked at trophic cascades in our last lesson, describing how predators within the ecosystem have multi-trophic effects. So they don't just affect the organism that they feed on, but also by default cascade they affect to other organisms such as plants, for example. And this very much influences both above and below ground relationships. And this is a very incomplete soil food web diagram, but it shows sort of the key players within the soil ecosystem. Of course, there's way more interactions than this shows. We've got our organic residues, the different bacteria and fungi that feed on those residues. And then we've got protozoa and the multicellular organisms that feed on protozoa and bacteria and fungi. And they are lumped here together as the microfauna based on their size. But of course, these guys, the protozoa, are actually much smaller than the nematodes and the rotifers. And the nematodes and the rotifers actually feed on protozoa. So they're a very good food source for these two groups of organisms. Size-wise, they are much smaller than the rotifers normally. It's rare that there are protists the size of rotifers, and they are very much a food source for both rotifers and nematodes, as well as the larger organisms. And this diagram is trying to represent the energy flow from organic residues up the food web and how it's distributed via the primary level consumers, the bacteria and the fungi, and then the secondary level consumers, so the second trophic level and so on. So it's really cool to see it this way as well, because we get this representation of how the plant foods, with the addition of nutrients that fungi and bacteria pick up from soil, then together get distributed up the food web all the way to us humans and other big herbivores and predators. And what isn't shown here is that all the waste from all these organisms, everything that they release as excess nutrients from themselves, becomes plant available nutrients. And that's key, because that is exactly what then drives plant community structure and diversity, as it modulates the soil environment for those plants to grow, as the soil structure as well as the soil moisture and everything else is very much dependent on all these organisms being present, constituting a healthy soil food web that then supports plant growth and diverse plant communities. So we have covered in detail the essential role of soil protists in nutrient cycling, as well as the benefits to plant growth, how that affects below ground and above ground ecosystems. But soil protists also play other essential roles in the soil ecosystem. And one of these is that they serve as really good bioindicators. So this is where we use biological systems or species or groups of organisms as indicators of environmental state. So the health of the environment based on particular biological activity. So soil protists can be used to monitor ecosystem restoration efforts. They can be used to review fire history. They can be used to see environmental pollution or the reaction of the ecosystem to environmental pollution. We can use them to test water quality, as well as soil and atmospheric pollution, and even climate impacts can also be studied using soil protists. And there are 7 particular reasons why protists are great bioindicators. And this includes environmental sensitivity. So they're relatively sensitive because they don't have walls around them. So they're permeable and they feed on a lot of smaller organisms. So therefore, those organisms would potentially have pollutants in them, and the protists then eat them, and they could either be sensitive to them or could biodegrade these. And they have a very significant functional importance in the soil ecosystem, being the primary consumers of bacteria and other microbes. Also, they are widely distributed across the globe. Many species are global, but also their distribution is pretty even across soils and very much dependent on the specific soil ecosystem. So ecosystems can be compared based on the presence or absence of particular protists. Their size and numbers are wonderful because they're tiny in size, so that makes them easy to collect and easy to monitor. And as well, being ubiquitous and being present in large numbers allows us to easily implement them as bioindicators. They have a very quick response time, so their population growth is very rapid. So that means that it's easy to see what is happening within their populations very quickly, and that is a very positive thing when you're trying to assess environmental impacts. For example, otherwise you might be waiting for birds to breed over time, or elephants to breed, or plants to grow. But soil protists can give us this really quick check as to what is happening in the ecosystem. They are easy to analyze. They can be either grown in the lab, but most likely these days their presence is identified and examined using molecular techniques. And some of these soil protists, particularly the tested amoebae, have great preservation potential, having shells and being able to be left in the fossil record, for example. So we can see over time what has happened in the environment based on these protist shells as bioindicators. So one of the most pertinent ways in which soil protists are used as bioindicators is in ecosystem recovery. There is this fabulous project in China called Green for Grain Initiative, where damaged parcels of land that have been under cultivation and have experienced erosion, soil salinity, and desertification, for example, are being recovered, where the ecosystem is allowed to regrow and people plant native vegetation on those plots and try to regain soil function. And this particular study has used soil ciliate community changes in examining the efficacy of the Green for Grain program. So they looked at different land parcels, some that have been under recovery for 15 years, some 13, some were just transitioning, and some plots were still under cultivation. And they found that ciliate community patterns were very much dependent on soil water content, soil porosity, soil organic matter, total nitrogen, and vegetation diversity. So all of these factors were correlated with ciliate community patterns, and these factors had the greatest impact. It is worth mentioning that nitrogen fertilizer is another great disturbance that actually significantly impacts phagotrophic protists. And this is in another study that was done specifically on the effects of nitrogen fertilizer on the soil community, finding that the very organisms that are supposed to release nitrogen from bacteria in the soil actually end up being removed from the ecosystem. And this total nitrogen here actually just means nitrogen that develops naturally over time with plant succession. Very different to adding nitrogen fertilizer in a nitrate form and actually killing the organisms and sterilizing the soil. So it's very important to understand that this cultivated land is the problem, the way it's being farmed. And it's really important to know that if we remove this fertilizer component in land management, these phagotrophic protists will actually take that role on and provide the natural nitrogen that is gained by bacteria in the soil. So going back to the study now, this particular ciliate community pattern was examined by studying species number, individual abundance, and diversity indices of the different ciliates. And all of these have shown that where the land was under restoration in the Green for Grain initiative for the longest period of time, that's where these positive ciliate community parameters were most significant in those parcels of land that were under recovery for the longest period of time. And the values for these parameters decreased as the recovery time decreased. And of course, they were at their lowest when the land was still under cultivation. They have observed that ciliate community succession shifts were strongly correlated with the ecological complexity of the ecosystem. So the more biodiverse and the more mature the ecosystem became, the greater the ciliate community succession was present. So more biodiversity, more numbers of species, more abundance of individual species, and greater diversity of ciliates. So they concluded that soil ciliate communities are a fantastic ecological restoration indicator. So this is one way soil protists can be used in order to assess what is happening in the above ground ecosystem. Is it healthy? Can those trees be supported for longer periods of time? Is the ecosystem recovery going well based on what is happening on the micro scale in the microcosmos of the soil using soil protists? Soil protists also play an important role in bioremediation. So bioremediation is when biological entities, that means any living thing, is able to transform or reduce the concentration of a particular pollutant in soil or any other parts of the ecosystem, like water, for example. Phytoremediation is the same, but it happens specifically with plants, phyto meaning plant. And the kinds of pollutants that can be transformed, remediated, removed, or reduced in concentration include heavy metals through binding, like happens a lot with fungi, for example, where their cell walls can absorb heavy metals. And there's other ways of heavy metal entrapment as well. And also, of course, organic contaminants of all kinds, like oil spills, and more things like microplastics or like nanopollutants are becoming an issue as well. There are some plants that are actually hyperaccumulators, so these have large root biomass and can, for example, absorb heavy metals and like having heavy metals in higher concentrations in their tissues as opposed to other plants. So there are many examples of plants that actually hyperaccumulate heavy metals. But more often than not, bioremediation is multifaceted. That means there's an interaction between the plants and the microbes. And a lot of complex pollutants require this entire ecosystem to function in remediation. The greater the amount of exudates, or the greater the concentrations of exudates that a plant can deliver to the soil ecosystem via its root system, increases microbial growth and metabolism. And this greatly encourages degradation of organic contaminants. This is, of course, plant species dependent, nutrient levels in the soil and the nutrient transformation within the soil, the root physiology of the plant, the overall environment, such as moisture content and temperature of the soil, and of course the microbial community which the plant species supports. But protozoa in themselves have a great impact, as we already know, on plant and microbial growth, which stimulates production of root exudates by plants. Protozoa are also the primary grazers of organic contaminants degrading bacteria, and the presence of protozoa consuming those bacteria greatly increases contaminant degradation. An example is when the flagellate Heteromita globosa increases the degradation rate of benzene and methylbenzene during the logarithmic growth of the bacteria that degrade these contaminants by over 8.5 times. So if the flagellate is not present, there is a lesser degradation rate by 8.5 times. So that's a huge increase in degradation as a result of this predatory behavior on the benzene and methylbenzene degrading bacteria by the flagellate. Also, toluene biodegradation is increased by Pseudomonas bacteria when Heteromita globosa predates on the Pseudomonas. So this is just an example showing the mechanisms of bioremediation, how pollutants can be cleaned up, and the vital role of protozoa in the bioremediation process. There is a really good review that came out recently that examines the interactions between soil protists and pollutants, which is still an unresolved puzzle. And that's because protozoa are often unaccounted for in their role in bioremediation, and there is very little acknowledgement of soil protists in general in the soil food web. However, there are several mechanisms that soil protists engage in the bioremediation process. And bioremediation can occur through various ways. So there could be bioaccumulation, so where an organism actually accumulates, like the hyperaccumulating plants, accumulates the particular pollutant. There could be biosorption, where a particular pollutant such as heavy metal might be adsorbed to the external structures of a particular organism, such as fungal hyphae or roots, for example. There's also the process of biotransformation, and this is where the pollutant is actually transformed, broken down, and made into something else. Or there are particular chemical structures added to it and it makes it no longer toxic. There's also extracellular chemical precipitation. So there are a lot of different ways in which different pollutants may be biotransformed and in which bioremediation can occur. And it's largely dependent on several factors, and that includes nutrient mineralization. Is there enough nutrients to actually support bioremediation? Of course, bioremediation is an energy and expensive process for organisms, so there needs to be enough nutrient cycling in the system and enough mineralized nutrients in order to support the organisms that perform bioremediation. And so therefore, this is where the soil protists, particularly the grazers or the predators, really affect bioremediation because they are the primary nutrient mineralizing organisms. They also activate bacteria, and that includes stimulating their reproduction as they graze on bacteria, so increasing their quantity, consuming aged cells that are likely to have a lot of pollutants in them in a polluted ecosystem, and also release active substances that stimulate bacterial metabolism. Selective grazing is another way in which protists affect bioremediation, because they might reduce competition and increase populations of pollutant-degrading bacteria. Being motile organisms, protists cause physical disturbance in the soil, so they actually increase oxygen content in the soil and the substrate surface area, so that makes it more likely to be biodegraded. So just by the protists physically mixing the soil and making space in the soil for oxygen and water to enter, this mechanistic movement allows for increase in bioremediation potential. There are also specific enzymes that some protists produce that have a direct degradation capability. So the protists themselves are actually capable of degrading the particular pollutant. And also there is symmetabolism, where bacteria, fungi, and protozoa work together to decompose particular pollutants. But also protists themselves increase energy and carbon availability for degrading fungi and bacteria. So bioremediation depends really on the grazing and metabolic secretions of protozoa and their effect on microbial community structure, and then also on the plant community that is supported by the protozoa action in the soil. And this affects root growth and the biomass of roots. And of course, in the end, it really affects the soil organic carbon pool. And this is very important because the more spongy the soil, the more complex, the more bioremediation potential it has, as pollutants can actually adhere, and the different biodegradation products can also adhere to the soil organic carbon, making it immobilized and therefore less toxic to the entire soil ecosystem in that area. This is a lovely diagram showing those interactions between soil pollutants and soil protists. So there are all different kinds of pollutants, such as microplastics, heavy metals. Organic pollutants and soil pathogens are also classed as pollutants in this review, and they are toxic, and they can be toxic to certain species of protists and other organisms, such as plants and bacteria. They can exert their toxicity at the community level or at an evolutionary level, for example, by causing mutations and therefore evolutionary adaptation to that particular polluted soil or to that particular pollutant. And soil protists themselves then interact with these pollutants and transform these pollutants if they are not killed by the toxicity of those pollutants. And this could be via accumulation, where the protists actually imbibe or take in these pollutants and are able to withstand certain concentrations, and therefore take some concentration of those pollutants out of the soil ecosystem while they're alive and hold on to those pollutants. They can also transform them biochemically through enzymatic action, cause changes in the chemical structure, and therefore rendering that pollutant either less toxic or even more toxic in some instances. And also through predation of bacteria and fungi that actually accumulate these pollutants. So by predating on the organisms that accumulate them, that then leads to the other two possibilities of biotransformation, but also release of the pollutants that are locked up in the bacteria and fungi. So there's a constant flux that happens via the protists in the soil of pollutants as a result of those interactions between the soil protists and the pollutants themselves, or the soil protists via the various other organisms that interact with the pollutants. So there are a lot of different, very little studied mechanisms that we are yet to understand, and this is why the article says that it's an unsolved puzzle that needs to be investigated further. And there are still many research gaps, and these include hidden diversity of soil protists. So soil protist diversity studies need to elucidate what is happening in the soil ecosystem in terms of the biodiversity of protists. What are their adaptive mechanisms? How do they actually adapt to these chemical changes produced by pollutants? What are the species interactions between the various protists themselves, but also with their prey and plants and all the other organisms that actually inhabit the soil ecosystem? They need to be studied as to their importance as bioindicators in the soil ecosystem, and we have touched upon that in the earlier slide. And also, how can they be used? What are their environmental applications in order to fully appreciate their bioremediation powers and their role in bioremediation of polluted soils? So this is it in a nutshell when it comes to protozoa and their effect on bioremediation. So let's just now summarize what we have learned about soil protists and look at some of the future research that needs to be implemented in order to further understand these incredible organisms and their role in soil health and soil restoration. So soil protists have a profound effect on the interactions between plant roots, root exudates, and microorganisms. In fact, they are key regulators in rhizosphere microbial processes and plant growth. And this is what we have already learned over the past 10 lessons. But the complexity of the various trophic levels is quite extreme, and there is a lack of studies. So long-term studies need to elucidate what are those interactions, how do soil protists fit into the complexity of the various trophic levels in the soil ecosystems. We also don't really know about the chemical signal transfer between soil protists and microbes. What is happening there, and how is this regulating gene expression and therefore drives the evolutionary processes within the soil ecosystem? The other not well understood phenomenon is that of the trophic cascades between underground or rhizosphere soil community and their above ground biota communities. How does the soil ecosystem interact and affect the above ground community of organisms, and vice versa? What does the above ground community of organisms, including plants and animals and insects and other life forms, affect the soil food web, that underground biological community? By studying these interactions, we'll be able to understand on a deeper level the rhizosphere ecological functions and soil ecology. We also don't know how climate change impacts rhizosphere ecological functions and soil organic carbon contributions, and how this affects the stability of global ecological functions. This is completely new territory, as climate change is having a huge impact with droughts. Protists can't live without moisture in the soil. They can't live without plants above ground, feeding the bacteria, the very food source that protozoa feed on. There's a huge and enormous question mark around what happens in that microbial below ground community as a result of climatic pressure that we're creating and constantly affecting all of the biological systems of the earth. So these are many questions that still need to be answered. And at the same time, as you have learned, it's already such an incredibly complex topic. So there are still many questions to ask, but I hope that you got some understanding now of the vital role of soil protists and how they unlock the nutrients from bacteria and fungi and therefore drive ecosystem function. Their roles are so key and so vital to the soil ecosystem. So I hope you got to understand a little bit about just how crucial these organisms are and how little we still know about them. So now that we have completed Unit 6 on protozoa or soil protists, we are moving on to Unit 7, which is about soil nematodes. Lesson 7.1 will introduce nematodes as the fascinating creatures that they are, and you'll be hearing from me again in Lesson 7.1. Bye for now.